URL to live. Use this link to live to watch. Use the rolling. Watch the webinar on YouTube Live. YouTube whatever they can.
Okay. And then
ठीक है से लिंक के साथ मोटी बोल रहा हूँ चेक करती ओके लिंक एड्रेस बारे में
छिबो ना मैं पठेई दिलु बे Okay.
Situation 83. Start one.
Okay, very good morning to all of you. I am Dr. Dilip Kumar Vej. Welcome you for this today's webinar. Today's webinar is on recent advances in breeding of commercially important fish species. So I welcome to the inaugural session to everyone, and I am so delighted to have all on board here. Welcome to the important session. We are going to talk about the recent advances in breeding of commercially important fish species. So friends, a wise man says, any science being pursued, if it is not relevant to society need, then it is just an honorary science. Again, I am repeating, any science being pursued, if it is not relevant to society need, then it is just an honorary science. We do not have that kind of luxury to have honorary science, at least in India. Science and technology always respond to society needs and global challenges. You know, science and technology, that is it is dedicated towards the society, what the society needs. So you know, the progress or advance, this is the key term to for our generation. As we know, rapidly growing sector of agriculture is the aquaculture. Aquaculture is expected to provide almost two-thirds of the fish intended for the global consumption by 2030. And aquaculture involves that breeding, rearing, and harvesting of fish, freshwater fish or marine water fish. So, as the world population growing, the scientists and researchers, policymakers, farmers, all are seeking ways to meet future food demand. Interest in aquaculture production, it is one of the solution to face these challenges coming ahead. So, the growing of agricultural based, that means growing demand of food needs, it is helpful in other aspect, that is improvement of the socio-economic status of the poor people and providing the consumers to high nutritional value of fish protein as compared to the other meat proteins, because the fish protein is very essential for the our health, because fish is containing the omega-3 fatty acid, polyunsaturated fatty acids that are helpful for our body's health issues. That means to prevent the coronary heart diseases, and the diabetic people also can consume this fish protein as compared to other animal proteins. So scientists developing advanced technology to meet complex agricultural challenges faced by throughout the world. They are cutting edge research to help farmers to produce higher quantities of safer and better quality of food to meet the needs of the growing population. So advances of the biotechnology have opened new options for farmers responding to market needs and environmental challenges. So production technologies which is developed in the biotechnology lab, that should be communicated with the farmers, because farmers are the smarter than the scientists. Farmers are actually the field officers. They recruit the manpower there. So they are very much smarter than the scientists. And biotechnology given us many things. Your chromosomal manipulation, production of the triploidy, tetraploidy, and hybridization, hormonal treatment for the sex reversal piece, induced breeding, and marker assisted selection for the to assess the genetic stock, and uniparental population is formed by the androgenesis, gynogenesis. Transgenesis is a significant contribution of the biotechnology, that the foreign novel gene can be introduced into the particular host. So to perform the check whether that gene is coming function by the gain of functions or loss of function, that means gene knockout technology. And is a conservation of the gametes, cryopreservation of the gametes. So that the biotechnology has given us many things. So today we are going to witness our honorable speakers from NBFGR, National Bureau of Fish Genetic Resource, and DCFR, Directorate Cold Fisheries Research Center. These two class of the high level ICR Institute. You will realize how much progress has been done in aquaculture our world has made, and that too more specifically in the fish reproduction, to enhance brood stock, preservation of genetic resources, etc. So ladies and gentlemen, It gives me immense pleasure to welcome all of you to today's webinar. I am introducing you the scientist who leading in India in fish reproductive biotechnology. All of you know the entire India is passing that two storm. One storm already passed and second storm is going on. So that is COVID-19 pandemic. Second wave is going on. So I wish all of you be safe. Take care of yourself and your family. I am grateful to all of you to attend this webinar. You took your time attend this webinar. All you deserve your heads up from me. And before we going to the topic, I would like to request Principal FM Autonomous College, Dr. Sangita Sahu, for welcome address. Madam, please.
Guest introduction टा दबो ना Good morning, everybody. Let me congratulate our invited speaker, Dr. Sulip Kumar Majhi, Principal Scientist, National Bureau of Fish Genetic Research, Lucknow, and Dr. Abhay Kumar Giri, Scientist, Director of Cold Water Fisheries Research, Bhimtal, Uttarakhand. Today, we are going to talk about a wonderful seminar, the topic being the recent advances in breeding of commercially important fish species. I welcome all the faculties, research scholars, and students who are pursuing their professional career in biological science. Today's seminar is highly necessary as it has very much impact on the agriculture. Demand of fish, as you know, is soaring worldwide. India, with an annual fish production of about 11.41 million tons. And this stands second among the top 10 fish producing countries in the world. But the increasing demand cannot be made through natural harvest only. Advancement in breeding could help meet the soaring demand. Modern technology in aquaculture and breeding have been made in the recent past in increasing fish production through various genetic and biotechnological tools. The potential areas of biotechnology in aquaculture include the use of synthetic hormone in induced breeding, molecular biology, and transgenesis. The future of fish breeding and aquaculture potential has been the subject of extensive discussion. Today's webinar will highlight about the application of biotechnology in fish reproduction and culture of cold water candidate species. Many thanks to the Zoology Department to conduct such a national level webinar. So let's enjoy the seminar. Hope this webinar will be fruitful and expand the dimension of your knowledge in aquaculture. Thank you all.
Speaker check
ଏବେ 100 ଆସିଗଲେ ସବୁ
speaker देखिए
ଏ ବଆ
सर्च कर देखिए ऊपर
ये
सर्च ऊपर कर
सर्च करिए
ऊपर पूरा
ना तो नहीं
देखिए अंदर सर्च करिए स्क्रॉल कर देखिए मतलब फोन ମୋତେ ପଚାର ଜଏନ୍
ହଁ
କରିଦେବ।
ହଁ ଓକେ ହଉ
ଯେତେବେଳେ ଖୋଲିବେ ଏ ଦିଟା ଅନ୍ କରିବେ, ଏଇଟା କ୍ଲୋଜ୍ କରିଦେବେ, ଏ ଦିଟା ଅନ୍ କରିଦେବେ। ଅନ୍ କରି କରିବେ।
So, welcome everyone. So, our first honorable speaker, Dr. Sulip Kumar Majhi, Principal Scientist in Department of Fish Conservation, ICR National Bureau of Fish Genetic Resource. He constantly proved excellence in developing technology in fish product reproduction. He has started his career in the year 2003 and he is having 70 years of experience in research. He has handled more than 10 research projects funded by external agencies. He has organized many training programs and workshops to build skilled manpower in the field of reproductive biotechnology. He has acted as an invited speaker in conferences, technical symposium, workshops, etc. His expertise in conservation and propagation of endangered, commercially important fish species. Those are difficult to breed in captivity by stem cell technology. He has published more than 50 research papers in reputed national and international journals. He has got Best Scientist, Young Scientist, Excellent Research Award, and very recently, Outstanding Scientist Award from reputed organization. He is acting as an editorial board member as well as a reviewer in various reputed international journals. So now I will request to Dr. Sulip Kumar Majhi sir, please start your talk. Yes, I am able to.
କହୁଛନ୍ତି ଶୁଣାଯାଉଛି କୋଉଠି ସେଥିପାଇଁ ତମ ନାଁ କଣ କହିଲେ?
Sulip, SU.
ଭିଡ଼ିଓ ଅଡ଼ିଓ ବେସ୍ ହବ
এটা ভিডিওটা অন
କଣ এটা অন করে কোন ক্লিয়ার ভয়েস ইজ নট স্লো সাউন্ড
No, 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 no. No, no, no.
voice is very low. Very slow.
No, 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 your slide is not showing. Present screen କରନ୍ତୁ. It's present, but slide is not showing, no? screen is visible, but your slide is not showing.
সেই স্ক্রিনে সিলেক্ট করে সেটা সম্ভব
দেখা সেটা কোটা পৌঁছে দেখা দেখা সেটা আমি কি করব এটা
এটা কোন দিকে দেখা যাবে
ସେ ଅପସନଟା ଦେଖ ଏଠି ଗୋଟେ ଅଛି
ଏଠି ସାର୍ ଏଠି ପ୍ରେଜେଣ୍ଟ ସ୍କ୍ରିନ୍ କୁ ଗଲେ ସେ ଶୋ କରିବ ନ୍ୟୁ ଟ୍ୟାବ ଏଠି ନି ଆପଣ କଲେ ହୋଉନି ସେ ସିଏ କରିବା
ତାଙ୍କର କରିବେ ନା
ତାଙ୍କ ଦେଇ କୋଉଠି ମାରିବେ
ଲାଇନ୍ ଫୋନରେ କଥା ହଉଛୁ
ସେ କହିବେ ଫୋନରେ କହିବେ ମ୍ୟୁଟ୍
no I am just only you, not slide.
ଭିଡିଓ ଅଫ୍ କରନ୍ତୁ ଆପଣଙ୍କୁ ଭିଡିଓ ଅଫ୍ କରନ୍ତୁ ଏଇଟା ଆପଣ ଫୋନ କରି ଡାଇରେକ୍ଟ କଥା ହୁଅନ୍ତୁ କିନ୍ତୁ ସେଠି ଗୋଟେ ଅପସନ ଆସିବ ସ୍ଲାଇଡ୍ ଶୋ ସେଠି ପ୍ରେଜେଣ୍ଟ ସ୍କ୍ରିନ୍ କଲେ
ତମର ଦେଖୁଛି ତ କେତେ ଦେଖୁଛି
Professor Shetty, hello. No. So sad. Yes, please.
Mane question it is visible, but I want to slide. देखो देखिए
Yes. Now it is coming. Thank you, Sidhu sir.
ସ୍ଲାଇଡ୍ ଶୋ କରିବ
Yes. Slide show. Please enlarge your slide. Okay. Please continue.
ସ୍ଲାଇଡ୍ ଶୋ ଅପସନ ସେଠି କ୍ଲିକ୍ କରିକି ପ୍ରୋସିଡ୍ କରନ୍ତୁ
So just that means there is a slide show tab. Please enlarge that one slide. Yes. So everyone, please, all participants are requested to mute their mic. Otherwise, it is unable to listen here. Okay. So please, Sulip sir, slide show. Click on slides. Okay. Please continue.
ବନ୍ଦ କରନ୍ତୁ ଆପଣ ଭିଡିଓ ବନ୍ଦ କରନ୍ତୁ ବସ୍
Okay, sir. Okay, Sulip sir, continue.
ଫୋନରେ କଥା ହୁଅନ୍ତୁ
नहीं अब देख तो जानिए कि विशेष भी खोल दे हुआ बंद कर
ଆପଣ ଦେଖୁନ କୋଉଠି ଭାଇ ଫୋନ କରିକି ଯଦି ପଚାରୁଛନ୍ତି କ୍ୱେଶ୍ଚନ ଆସୁଛନ୍ତି ଏଟା ମିଳୁ ଅଛି ନା ରେସ୍ପନ୍ସ କରୁଛି ସେ ମନ କରୁଛି ମୁଁ ଗୋଟେ ୟୁଟ୍ୟୁବରେ ଦେଖୁଛି ମୁଁ ଭିଡ଼ିଓ ଜଏନ୍ କରେ
ୟୁଟ୍ୟୁବରେ ଦେଖୁଛନ୍ତି କୋଉଠି ଆରମ୍ଭ କରିଦେଇଥିଲା ନା ପିଲାଙ୍କୁ ଏନଗେଜ ରଖିବା ପାଇଁ ଏତେ ଫୋନ କଲେ ମୁଁ ଆସିଛି ମୁଁ ଜାଣିନି କଣ
ଟିକେ ଡିଲେ ଅଛି ଏଟା ୟୁଟ୍ୟୁବରେ 5 ସେକେଣ୍ଡ ପରେ ଆସୁଛି ତ ଫୋନରେ କଥା ନା
आवाज काय असतो
ଫୋନରେ କଥା
तुम्ही सुनते क्लियर
ପୁରା କ୍ଲିୟର ଶୁଣୁଛି
सीटे लोग भी सुनो देखो सुनते सुनो
से कोरी ना
असे पोहोचून कर
ଆପଣ ତ କାମ କରୁଛନ୍ତି ମାଇକ୍ରୋଫୋନଟା ଅନ୍ କରିକି ତାକୁ କହୁନାହାନ୍ତି ଯେ ସ୍ଲାଇଡ୍ ମାଇକ୍ରୋଫୋନଟା ଅନ୍ କରନ୍ତୁ ଦେଇ ସାର୍ ବାନ୍ଧିଲେ କହିକି ସ୍ଲାଇଡ୍ ସେଠି ନା ମାଇକ୍ରୋଫୋନଟା ଅନ୍ କରିବେ
माइक्रोफोन में जो चार्ट
ହଁ ଚାର୍ଟ ବସିକି ଦେଖିବେ
Excuse me,
ମାଇକ୍ରୋଫୋନ ଅଛି ଆମ ବୋଇ ଯାଉଛି ଟାଇମ୍
Slide change होगी।
सीरियल साउंड ହେଲା ଭଲ ହେଲା 5 ମିନିଟ୍ ମଝିରେ କଟିଗଲା ତ ମୁଁ କହିଲି ନା ଇଣ୍ଟ୍ରୋଡକ୍ସନ ଲାଷ୍ଟ 5 ମିନିଟ୍ କଟିଗଲା।
कहे स्लाइड टा चेंज होची स्लाइड चेंज हो मन तक कहे hello Sulip sir please change the slide no that is in the first slide okay so it is not showing in our computer others they also not viewing your slide just in the first page assisted reproduction in peace hello slide so no slide show the Sulip sir you have done the slide show no but slide is not moving just in a fixed position that means in first slide okay so
ସାର୍ କ'ଣ କରୁଛନ୍ତି ନା ସେଇଟାକୁ ସିଲେକ୍ଟ କରୁନାହାନ୍ତି ଯେତେବେଳେ ଦଉଛନ୍ତି ସ୍ପିଚ୍ ମାନେ ସିଏ କ'ଣ କରିବା କଥା ନା ସେଇ ସ୍ଲାଇଡ୍ଟାକୁ ସିଲେକ୍ଟ କରିବା କଥା ସେଇଟା ମିନିମାଇଜ୍ କରିଦେଲା ମାନେ ସେଇଟା ହବନି ଏଇଟା ଦେଖଉଛି
online
Hello, good morning, sir.
Hmm.
Salute, sir. Are you hearing me? Okay, sir. Is your that presentation screen is visible on your screen now? Okay, okay, sir. Sir, have you made it slideshow? That means, okay.
Right.
Okay, now, sir, it is coming.
It is coming. It is coming.
Select करियो ये करियो.
Okay, sir. Now you can continue, sir. Your slide is visual. Visual, है ना?
So से मानना थी क्या कौन से select करियो.
जो बड़ो एकदम क्लियर करिया कथा बड़ो ढंग ना.
Sir, now you can continue, sir. Yes, sir.
Yes. Mhm. Hmm?
This slide is going on the slide. Screen service हमारी होती है.
Hmm.
Screen shot. Screen.
Control shift इसमें से.
हाँ, वो मान लीजिए उनको क्या लगा. Six slide ना वो वाला slide को कंट्रोल. हाँ, ठीक है.
Hmm. Okay.
ठीक है.
Yes. 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 Yes.
हम कौन कौन बस में.
Yes, sir. Yes, sir. Continue. Continue.
नेटवर्क इसमें हम लोग लाइव ही रहे इतना तो. केवल चेंज हुई तो चेंज हुई तो. हाँ.
Problem है.
तो नेटवर्क इशू का लाइव ही रहे.
रही.
Hang here बोले ना. Rest ले.
Uh,
quite depleted. So what we did is we injected copium to busulfan at 2 weeks interval, and after 12 weeks we could get these effects are developed after 100 growth. The detail of this experiment you can find in the Animal Reproduction Science. It was published in 2005. Now, analysis method that we apply to see whether the treatment that we give to this animal were really active or not. Apart from GSI, gonadotrophic indexes, where we need to kill the animal and then isolate the testis and then see whether GSI is a proxy of maturation status of the animal. The lower the GSI, it implies like the cells in the testis or so to say in the gonad, irrespective of the male and female, they are declining. So it is one of the proxy approach to estimate whether these animals are moving towards to become stable. Apart from that, to make very sure of what we see through GSI is by through histological observation. Here we do the histological section and then see whether the cells are depleting or not. And additionally, Vasa genes, or so to say, apart from Vasa, there are so many genes also. Nano, the rocks, those are specific to the germ cell in the testis. This kind of marker can also be used as one of the factors to corroborate what you observe in the histological section or through the GSI can be one of the additional factor where you can confirm, okay, apart from these two, you can through molecular approach you can see or whatever you are looking at the histological sections and all are corroborated through molecular approach. So basically we apply this three kind of technology of late. Also we are developing some of the non-invasive approach without killing the animal through endomicroscopy, whether these animals can be detected, whether the cells have been efficiently depleted or not. That I think within a year or so we'll come out with the technology. Now this is briefly some of the results that we got after exposing the common carb to the chemical and thermal treatment protocol. As you can see here in this slide, after end of 10 week treatment period, so most of the animals, as though they were depleted of endogenous germ cells, one good news is there were hardly any changes in the body weight, because if we give a treatment where apart from our interest of observation, if the animal physiology itself get hampered, then probably that would not be best approach to continue. So here we observe like although we subject this animal to chemical and heat treatment, but the animals were accepting the food and all those things, and their body weight changes were not that much significant. But interestingly, as I said, there was reduction in the gonadosomatic indexes after 10 weeks. Once we subject this animal to the heat and chemical treatment protocol. And then through histological observation, as you can see here in top panel, there were control animal where you can see the intact germ cell profile in the testis. Opposed to that, in the bottom panel, as you can see, there were absolutely no sign of any germ cells in the testis of the animal. Now, this is same in the case with the female. As you can see here, opposed to the control that we are presenting in the top and the bottom, you can see like, there was absolutely no sign. Of course, there were a sign of phagocytosis. Phagocytosis is another very interesting topic. Maybe that could take another separate class to explain phagocytosis and all. The gist of this is like once any cell or any so to say any chemical compound that enters into a recipient gonadal environment, those cells, say to say somatic or Leydig cell, they have a tendency to eat upon those kind of cells, cell of the foreign origin. So same is the case like here. What is once we inject busulfan, there were certain animals where we could see cases of phagocytosis. Now this is the Bhasa gene transcriptome that transcription profile that we did through real-time PCR. Whatever we saw through GSI and histology were also corroborated to the Bhasa gene expression. Now this is what kind of a criteria that will come out from this study is like apart from when you see in the natural water body, so to say. Now once you go to the field and dissect some of the animals, how you relate those animals whether they are reproductively active or they have lose their reproductive competence. Now, this is one of the criteria that you develop once you go to the field. By looking at the gonad itself, you can get a kind of assessment whether this animal are still sexually reproductive or they have already lose the reproduction competency. So what we see here is once the animal lose their reproductive competency, you can see the gonads tending towards looking like thread kind of a structure. It's a kind of a black thread kind. So if it gonad appear like in a black thread kind of a structure, it means it implies this animal have already lose the reproductive competence. This is one of the implications that we suggest. Now, this is one of the very easy to use protocol that we use, isolating the cell through density separation technique to discontinue proper gradient. Now, these are some of the data that we got, apart from collecting the cell of our target, we make sure we label them with the fluorescent dye. This neutral dye is PK26. There are so many dyes. Apart from PK, there is also green fluorescent dye and all the things. They can be used as one of the markers because point here is, once you transfer the cells into the recipient, the main objective is you have to see whether the cell of your interest are really going to the basement membrane and they are taking the position so as to differentiate into the donor origin gamete. That is the reason we dye the cell, the donor origin cell, so as to make sure when you visualize them through microtomy, uh, these cells are really going and occupying the position of their interest. Now, this is typical about the cell transplantation. We transplant the donor cell under the anesthetic condition. Here, the animal, the apparatus that we develop in the bureau, it can withstand up to 40 to 45 minutes. The animal can be under the subconscious status, and very easily and very efficiently the cells can be transplanted through the urogenital papilla. Of course, like as I said before the transplantation, we add a few drop of different glue to make sure the cell that we transplant are going into the target organ. Now, this is, as I said before, through cryomicrotomy, we observe in certain time of period the cell that we inject into the recipient, whether they are really going into the basement membrane and taking the position. As you can see here, this cell is a goldfish originated stem cell, which is labeled with PK26. Uh, as you can see, opposed to some of the cysts which are vacant, like these cells were ablated or depleted using the chemical heat treatment. So the cell that we inject in this animal, some of the cell having the stem cell property, they have successfully migrated and colonized in the basement membrane of the common carp. And after a certain time of period, these cells start proliferating along the basement membrane. And as early as in 6 to 10, for more time, these cells have started differentiating. So this is one of the kind of evidence that you already see, always see by labeling the cell. So kind of it's a kind of evidence that whatever cell that we are injecting into the recipient are successfully differentiating into the you know end cell, and that is spermatozoa or oocyte. Now, same is the case in the female, as you can see here. In the female also, the cells initially they get distinct, subsequently they make a chain of the oogonia cell, and uh, after 4 months they differentiate into the donor origin oocyte. Of course, this is when you after these cells they differentiate and the animal get mature. We try to strip this animal to generate those spermatozoa and the oocyte. And prior to fertilization, we make sure to observe them under the microscopy, plus to get confirmed whether whatever the spermatozoa or oocyte that we get are having donor origin cell or not. This is one of the mechanisms that we argue after observing all those things, all the parameter in the first phase, the cells once we inject into the vulnerable environment, they rapidly get distributed. Subsequently, because the approach that we adopt here is very easy to use approach. That is through isolating the cell through discontinued proper gradient. Although, as I said before, also you can also use the flow cytometry and other high-end approach. But here, one of the concern here is apart from your cell of interest, there will be certain contamination of the somatic cell at there. We cannot ignore that approach. So, while injecting the cell into the recipient, there would be certain chances like we are also injecting the somatic cell. So the stem cell have only got the characteristic of having migration and colonization. So in the second phase, the cell having the stemless property, they will only migrate and colonize at the western membrane. And in the phase three, they will successfully differentiate to produce the donor origin cell. Now, this is one of the approach that you do always apart from artificial insemination. What you do through stripping the animal, We also make sure this technology works through uh, artificial, apart from artificial insemination, work through the natural spawning. So we used to couple the animal for the natural spawning and produce the progeny. Now these are some of the data that we generate from this crossing and all. This is again for the artificial insemination crossing. Now, another approach that I would like to quickly cover is one of the innovative approach that we developed in the institute is, stem cell therapy approach to reprogram the reproductive competence of the animal. Say, for example, we have a high and high valuable species, and you don't want to lose or don't want to sacrifice the animal for the sake like it has lose the reproductive competence. Here, stem cell therapy can be one of the approach where you can revitalize the reproductive competence of such animal. Now, this as you can see here, basically in fish, there are three phases of the reproduction in the animal. One is prepubertal, another is in pubertal life where the animal contribute generously to transmit their genetic material to the subsequent generation. And then there is, of course, one of the phases post-pubertal where they lose the reproductive competence. But our approach is to use post-pubertal phase also one of the phase where the animal still start continuing contributing to the contributing to generating their contributing their genetic material to the subsequent generation. So here, what we do is, we make a therapy of this animal, those who have already lost or those we call as a senescent. This animal can be given an approach of stem cell therapy where they again revitalize their reproductive competence so that until they die, I'm not sure because I haven't still recorded any result like until they die, but theoretically, until they lose their life, this animal genetic material can also be used for uh, producing viable genetic material, those we have already used in this type. So here we have taken a goldfish as a model. It's already 12 years old. So we did all sort of studies to make sure and to see whether these animals are having still reproductive competence or they have already lost. So we see through the histology and all. And we also isolate the cell. Actually, here there are two approaches. One is like you can use already the diploid cell in the sense like for the thin flipping and other things. Uh, the cell can be isolated and through cell culture techniques and all, this cell can be proliferated and can use uh, this cell for the transplantation. So what we did is through catheter, we isolate some of the somatic testicular cells from this animal, and we give the rapid cell through rapid cell culture techniques. We proliferate these cells, and we inject the same cell to this animal to reprogram the reproductive competence of the animal. Now, these are some of the data that I'm sharing with you. As you can see here, after 12 weeks, you could revitalize the innocent animal to produce those spermatozoa cells from this animal. We didn't try with the female. Of course, we will be trying this year using the female. Now, these are the sperm count data that I'm sharing with you. As you can see here, they were from here. You can observe after the therapy protocol, the cell count has drastically increased. These are some of the data about the breeding using the cell that we have based on the senescent animal after the therapy. Same again the animal reproduction. So another interesting thing is like people used to say like this paper you can see in the medical journal PJ, it is already published, it is in the public domain. So one of the reviewers they have asked like once you generate the cell after therapy, is there any abrupt changes or is there any abnormality in the embryonic development? So here absolutely we don't see any kind of changes in the embryonic development. They were absolutely at par with the normal embryonic development. So at the end, it can generate the functional gametes even after the animal attain lose their reproductive competence through stem cell therapy. So potential application of our technology that we are continuously developing in the bureau is apart from surrogacy, we can use this technology for transgenic also, and of course we have been using this for the germline conservation of the species which are rapidly moving towards become endangered or extinct. Thank you so much for your kind attention. If you have some questions, I'm happy to answer. Dilip?
So my question is, after random distribution of donor germ cells in seminiferous epithelium, a few migrate to basement membrane. So what happens to the residual germ cells?
Actually, what happened, as I said, the approach that we follow here is using the density dependent separation technique. That is using purple as one of the ingredients. So here, what happened apart from stem cells, there are also contamination of the somatic cells and other kind of cells. So these cells, which doesn't possess the stemness characteristic, they have been ultimately phagocytized. So they are being eaten up through the, and because one thing you have to remember here, those somatic cells are probably one of the toughest cells present in the body. So their duty is to protect the vulnerable microenvironment. When the sea kind of a foreign cell, particular the other kind of donor origin somatic cell or other cell, so they virtually end up eating those cells through phagocytosis approach.
Thank you, sir.
Sir, what are the disadvantages of stem cell therapy?
No, like as a, I'm hearing a lot of noise in the background. Okay, thank you. So to say disadvantage, I don't know how to categorize it as a disadvantage, but of course, apart from disadvantage, we have a lot of advantage because what we do is we extend the reproductive lifespan of the animal through the technology. So absolutely disadvantage as of yet, we haven't seen any kind of disadvantage. More of that, is only the advantage because we are harvesting the genetic material for an extended period. Thank you.
Thank you, sir. But in human beings, we are seeing stem cell therapy has lot of disadvantages, like it can cause lung fibrosis, like that.
No, actually, here is like the stem cells that we have is from where we are isolating the stem cell. That is one of the biggest questions. Say, for example, in human being, and so to say, in especially in the females, like those who are having some alignment of reproductive competence and other things. Now, question here is where we isolate the stem cell from where? It should not be from the exogenous origin. It has to be from the same individual, maybe from the skin or maybe from any other tissue that we have as the stem cell. Then there won't be any chances of immunorejection because immunorejection is another factor which we have to keep in mind. So in fish, what we do is we use the same animal cell from the gonadal environment and we multiply these cells and we label them and we transplant these cells into the target organ that is the gonad. So in human also, where from we harvest these stem cells, that's the biggest question.
Okay, thank you, sir.
Yes, sir. Uh, Yes, sir. Myself, Barsha Baisakhi. I have a question where I want to ask the success rate of this new therapy as compared to the traditional induced breeding.
Actually, see, therapy is one of the way, like, where you can extend the lifespan, as I said before also. But conventionally, you deal with the animal in its reproductive lifespan only. Say, for example, Rohu, Katla, Mriga, whatever it is, we breed this animal for 4 to 5 years. After that, it's gone. Cannot use the animal for the breeding purpose. But here in therapy, we are talking what about after 4 to 5 years? Whether we can use the same animal to generate the genetic material. Say for example, it's a high elite germ plasm. We cannot simply afford to lose the genetic material. So through therapy, we extend the reproductive lifespan so as to generate more of the genetic material from the same individual. So absolutely, there is no comparison between this approach. Once in one side, we are talking about simply induced breed the animal, and in other hand, we are talking about how to extend the reproductive lifespan of the animal. Thank you.
Okay, sir. Thank you, sir.
Thank you. See, my pathetic situation is like after end this, I have another meeting. So I don't know how to deal with all these things. Anyway, thank. I'm really thankful to you. I am thankful to FM College and convey my regards to Madam also, everybody who is out there for giving me an opportunity to explain my work. We are doing our best to safeguard the secrets of the country, and we will be continuing doing that. I'm thankful to every one of your participants who have joined the meeting. Thank you so much. Pleasure. Pleasure.
Thank you. Thank you so much, sir. Can you hear me, sir? Yes, yes. Yes, yes. Yes, yes. Thank you. Yes, yes. Can I share my slides? Yeah, na? This one. Is it visible, sir, now? Full slide mode? Thank you. Thank you. Thank you so much. Good morning to everybody. And it is very nice to interact with the peoples of Odisha, mass peoples of Odisha after a long time, because I often used to go to Odisha. Actually, first I want to introduce myself. I am Abhay Kumar Giri, and basically I am from Odisha, and that too from Baleswar. And so I have joined my ARS scientist in 2014. Then I posted in our some DCFR field center, that is Champabad. So my most of the views and observation will be on the farming and practical application with quick field validation. And for 5 years, I saw that Champabad is our field center, one of our field center that you can visible in my slide in the left side of the screen. That is serving at Champawat farm. Now I am back to headquarter Bhimtal, which is located around 180 kilometers from the Champawat farm. So now here I am 2 to 3 years. So this is the brief introduction of myself. Now I will discuss about the some of the Champawat, and that earlier this Champawat farm is generally used for the only for rainbow trout, and because we know in cold water scenario, in the hill farming scenario, rainbow trout is the most promising species. Of course, it is not an endemic or indigenous species. It is exported from outside India, from where from England, and introduced so many places in our country, like Jammu Kashmir, some of the high hill areas of Tamil Nadu, like Nilgiri, Uti, like that, and Himachal Pradesh. But during our time, and we conducted some of our rainbow trout ongoing activity, and also some of the indigenous species like snow trouts, because as we know, in our Odisha also, some of the nowadays, some of the small indigenous species that categorize as SIF, small indigenous fish species, and that are actually nowadays getting importance because of its high stocking ratio, and also it is very suitable to the environment that is we are culturing, and also it is having good breeding performance. Automatically auto breed, it can auto recruit its offsprings, and also having good quantity of minerals and trace elements. So for this purpose, we also continue to do some of our snow trout species that are available in our local stream. Along with this, some of the exotic varieties of exotic carps, a common carp, the breeding and seed production technology we are basically doing at Champawat. So now I will discuss about what are the basic outline of the candidate cold water species and their farming practices, and that is going on in our hill regions, and what are the significance. So first we will see the fish diversity. What are the diversified species that are available in our hill regions? Actually, our hill region in cold water, our hill region, then basically a huge area. Basically, huge area. It includes 7 sisters of northeastern states, then Sikkim, all the way to Jammu Kashmir, Leh Ladakh, Himachal Pradesh, and also including some of the southern parts like Munnar hills of Kerala, then Nilgiri of Tamil Nadu. So it is a wide area of the cold water species to be cultured and to be explored. So, as I discussed, some of the fish diversity that are available in our cold water regions that includes snow trouts. Snow trout, basically, it is a very small indigenous species. It can go up to large size like 1.5 to 2 kilo, but it's a long duration. But basically, in our local streams, and the small varieties up to 150 or 200 gram that is available. The growth rate is very slow, but people generally used to supplement this species along with some of the car variety due to the local market. Generally, snow trouts are coming within 6 genera like Schizothorax, Schizopilgopsis, and some of the Lepidopilgopsis, some of the Diptychus species. These are some of the genus, and among the genus, there are 17 species of snow trout. These are some indigenous species, and another one is Mahasir. Mahasir also belongs to generally three genera. These are actually very high valued hill species because due to its, we know, due to some of the anthropogenic activity, some of the climatic pressure, and also some of dam construction for hydroelectric project, and many of these. So these species are the breeding ground. These species and the population of these species are declining day by day. So our motto, our aim is to protect these kind of our valuable species because IUCN also categorized the Mahasir species, basically the two Torputitera, endangered species. So Mahasir basically belongs to three genera, and this is Tor, Neulosacillus, and one is Nazirita. So basically in our area, Tor Putitera, Tor Kudri, and some of the other test species are Tor Musala, some of the other test species also available. And we also might be knowing in our Mahanadi also, Mahanadi River of Odisha, the Tor species, Tor Mahanadiscus also is available. So, because this species has a wide range of thermal regime, it inhibits in our middle regions and also in some of the foot area. Similarly, some of the Neolophilus hexagonalapis, Hestasticus, Strachi, some of the Neolophilus genus is available, and only one species is available in our Nazirita group, that is Nazirita chelinoids, that is in dark Mahasir. And including this, some of the exotic varieties that I was talking about, some of the exotic varieties like trouts, Oncorhynchus mykiss, that is rainbow trout, some Salmon putrafario, that is brown trout, and some of the char species like Salmonellus. These are the available, these are actually available some of the European and England the native species, but these are actually way back in 90s. Is introduced in India due to their first introduced due to sports fishing activity, and seeing their cultural importance, their growth status, it is included in our culture practices. So some of the other exotic species like our exotic carps, exotic
major carps like silver carp, grass carp, and common carp. And common carp also 3 varieties are available. And apart from this, some of the minor carps that nowadays we are more focusing about the minor carps, and because people generally prefer minor carp for their culture, and also it can be stocked in high density, and it is having local market in higher price, because if we'll sell the some of the IMC or exotic crafts around 200 or 250 rupees per kg, and some of the minor craft we can go up to 600 700 rupees per kg. So some of the minor calves, Pero, Dichelus, and some Garagotyla, some of the Semiplotus, more than 15 or 16 species are available that in our cold water region to be cultured in our valley and some of the foothill region. And along with this, some of the indigenous species like barrels, minnows, some sucker fishes, some catfishes, loaches, algae eaters, these are having more or less culture-based fisheries contribute, and also these are generally included in our ornamental varieties. So all total, generally 278 species are available in our cold water regions, and in Himalayas region, this is actually basically demarcated into Himalayan and Deccan Plateau region, and more are found in Himalaya region, 203 species, and 91 about 91 species in Deccan Plateau region. As we see, the seven sisters of the northeastern states, these are known as seven sisters: Arunachal Pradesh, Assam, Meghalaya, Tripura, Mizoram, Manipur, Nagaland. These seven states, these contribute maximum diversity of cold water species, and among these states, Assam generally contribute. Assam generally has the diversity of more number of species, to 18 species, followed by the Arunachal Pradesh. What are the what are the species that they are culturing in our cold water aquaculture? In cold water aquaculture, basically in cold water region, we demarcate cold water region into three categories. One is foothill region, middle hill region, and another is high hill region. Foothill region generally it is demarcated up to 750 or 800 meter altitude, and from 750 to 1500 altitude it is middle region, and more than 1500 altitude it is high hill region. And in foothill region, some monoculture of the carps, exotic carps, and some of the IMCs like Katla, Rohu, and even nowadays people are culturing Pongas variety, the what is that? The Ponga species Hypophyseton, that I actually scientifically I am forgetting, but Ponga species they are actually culturing due to the demand, because after 2006 the Ponga species are generally banned due to the some of the invasive nature, and it can some destroy some of the natural fish fauna. But after so many meetings and so many alterations, so many high level meetings, they actually, the government has lifted the ban against this Pongas. So nowadays Pongas also nowadays culture in some of the mid hill and also foothill region, and in high hill region we are basically targeting about the rainbow trout, because it is a high growing species and also having cultural importance. And farmers are practicing these species due to some of the export market. Not only the export market, in locally also it is getting a high price, about 500 to 600 rupees kg. And some of the big hotels, like it is also now we are also transporting this fish to Delhi and Mumbai. Some of the big hotels we have a contract. So, and they are actually in some of the farmers, or some of the, we can say some of the entrepreneurs, they are also selling directly in Delhi about more than 1500 rupees per kg. So along with this, that I have discussed some of the potential species like Richardsoni, Niger, some Progastos, Plagiostomus, and some of the Mohacis species, and some of the carps and bar species like Dero, Ditile, Semiplotus, and Belangiri, some Choconius. So we'll discuss later one by one. So first will come the basic species, that is rainbow trout, Oncorhynchus mycis. It is basically a salmoniform, and this is in salmoniform. Some of the are the sea-going form, some of the landlocked form, and this is the landlocked form of the salmoniformis. And it is basically cultured in our freshwater areas, unlike the some of the salmon, it is cultured in marine areas. Marine, and it is restricted to cold water region because its thermalism is around 18 to 20 degrees centigrade. It can grow well, and more than that, some problems may occur due to its feeding expectancy, its growth rate, and basically it is a very lucrative species. You can see the color of the lateral line. Lateral line color, it is giving some rainbow-like appearance, and that's why it is given rainbow trout. So now we'll see where it is originated and how it came to India, and what is the prospect of this rainbow trout. So basically we see, it is very native to North America. And it is farming started around 1853 in North America, and in Europe. Then it is first successfully transferred to from North America to Japan, and later to England and Scotland in early 80s. And here I wanted to mention one of the things. Unlike we know some of the most of the species, unlike carps and catfishes, we generally transport the fry, spawn, or fingerling size. Unlike the rainbow trout, it is some of the developmental stages of egg called eye. This eye is most suitable for transportation, even from foreign countries and within the various states of our Indian constitution. So global trout production. It is nearly farmed 82 countries in the world, and it is some FAO data in 2014-15. It is exceeding 855,000 ton in a global scenario. And Chile, you can see Chile 33%. It is the trout production. But India now, it is very in maker state. It is in the very low state to be expressed in global scenario. So then come to the Indian trout farming. Indian trout farming, and first attempt in introduce the eggs that I was talking about, the Idova from New Zealand. It is early 80s, and Dr. Sir Francis Day he introduced in Nilgiri. Nilgiri, it is the you might have heard about the Tamil Nadu Ooty Lakes, and near the Ooty Lakes, Nilgiri is there. There it is introduced. Then it is transplanted to from Europe. Basically, this is a sport and game fish. Means people love to play this with this fish because it takes bait very easily, and it will fight during your angling. It will fight very enormously. So people love that nature of this fish. So basically, it is introduced in our India for the sport fishing, recreational fishing, ecotourism, angling, this kind of purposes rather than culture practices. And seeing this kind of activity and this type of scenario, first rainbow trout hatchery. It is trout hatchery means first brown trout hatchery actually. Brown trout hatchery. It is established in Harwan, Srinagar, near Kashmir Valley in 1901. So, what is the ICAR-DCFR effort during the introduction for the introduction of the rainbow trout? In DCFR Bhimtal, as I mentioned, I was working in Champavat, so it is early. It is 1999. It is introduced in Champavat, taking the Ayodhya from Himachal Pradesh, and then since then it is distributed to farmers and different state departments, and successful ICR DCF are also done successful breeding and culture of rainbow trout in some of the states like Uttarakhand, Sikkim, Arunachal Pradesh, Nagaland, and recently I went to some Munnar hill of Kerala and also there we have standardized the seed production of rainbow trout, because in Munnar hill of Kerala it is very old farm. It is British level farm. It is early 80s it is established, but it is not in such a good condition. So we have standardized how we can produce and seeds and for the local farmers, and because some of the cold water region local farmers in that Munnar hill area, they have interest in culture and breeding of the rainbow trouts, and to revive the old stock and to have the more growth rate and more performance of the rainbow trout. So recently we have introduced about 2.5 lakhs of Ayodhya from Denmark to ICRTCFR to Jammu Kashmir State one Budgam farm in Jammu and Kashmir. So we have introduced the Denmark stock at Jammu and Kashmir. So now we can say why we should go for trout farming. What is the benefit of trout farming? We might have know that generally some of the carp farming and trout farming, this is some very means antagonistic scenario, because basically we can see we can supplemented farming. It's in the sense it is not completely feed based farming, because in trout farming we supply the feeds to the stocked fishes. That has the we can say it is the supplementary feeding, because some of the fish foods that are generated inside the tanks in the form of plankton, that is phytoplankton and zooplankton. But in case of trout farming, it is a completely feed based system. So it is easy for domestication and no culture problem, unlike carbs, because in carbs some of the we have some of the plankton that is having some good as well as negative effect also, because during blooming season time, especially during in our rainy season, some of the bloom occurs of the phytoplanktons, which means increases the DO level abruptly during the daytime, but subsequently it is very alarming situation during the night time, which affect and also cause the death of the farm fishes. But unlike the carbs, we can farm trout, or rather than we do have some clear water quality. So basically these species directly consume the feeds, and based on the feeds their growth also occurs. And also nowadays we can this fish offals from the trout farming has a good nutrition value, having good amino acid profile, and some of the protein, good percentage of proteins, and some of the high quality lipids it contains. So it can be used by the feed industry, and also it has the faster growth. And unlike cars, we do have some breeding protocols like injection of the our brooders or the next phase of the offsprings. But in case of trout, no induction is needed. It basically based on the feed quality and also the environmental parameters and the season. And It is but I because it is carnivorous, so accept artificial feed very properly, and wastage of feed also very less, and no need of manuring and fertilization like in case of calves, and very tasty with less bone content, means high muscle to bone ratio, and good nutritional profile, and it has the good potential of revenue generation as well as for local market and also for international market. And some of the farmers, they actually some complete structure they used to develop for farming as well as for tourists, for homestay, and also some angling facility. So it is a complete package of practice for the earn and for the economic upliftment of the farmers, and it is also having the post harvest and value addition. And that I was talking about the what is the direct income source, that we can make a cold chain for transport to some of the high-held hotels and metropolitan cities. Some of the angling status, we can earn also some money from angling, because from foreigners in our Bhimtal area and Nainital area, and also Jammu Kashmir and Himachal Pradesh. Some of the hill areas, they generally attract some of the Indian tourists and also foreign tourists. They used to stay, and for angling rod, they used to pay per day about 400 to 500 rupees. Means without sacrificing or without the means without your sacrifice of your fish, you can earn the money by playing the tourist with the fishes. Then now we will consider some of the trout farming, where we can construct the trout farms. What is the selection criteria? That I was discussing about this. What is the site of the trout farm? It is basically the permanent clear water source, and it is while we are talking about the water source, basically in hill regions, two types of water source is available. One is spring fed area, another is flood area, means flood water coming. So when excess rain will be there, then the catchment area, the river catchment area full of the water, and this water also we can add for the trout production. But some of the Jammu Kashmir area, they are having spring fed water. Spring fed water, it is the basic beauty of spring fed water is it generally does not fluctuate so much. The temperature and water quality are more or less similar across the season. And the also trout water should be free from the environmental pollution, heavy matter, and also turbidity, and the thermal regime around 5 to 20 degrees centigrade, unlike the parks. And DO should be 80% saturation up to 9 ppm or 7 to 8 ppm, and pH also slightly alkaline. It is preferred as of the many of the fishes, and it should be. And during the construction of our trout farm, the farm site should be 1 to 3% of the slope. About 5% slope, so it is good, because when we are constructing the trout farms, trout generally culture in flow through system, means one end the inlet water are entered, and another end it is outflow of water. Unlike the carps we can culture in stagnant ponds, but in trouts, so with the for compromising or for not including the cost of the pumps or pumping system, we can have the gradient flow, gravity flow of water from one end to another end. And of course, accessibility to uncollected road and also nearby feed mill and also marketing facility. For rainbow trout farm, basically two types of rainbow trout farming can be done. One is from hatchery phase, that is from breeding and up to rearing up to fry or fingerling size. Then another one is grow out stage. In hatchery phase, the optimal thermal regime around 9 to 11 degrees centigrade, slightly lower but the lethal for the incubating eggs and our fries more than 14-15 degrees centigrade is lethal. But during our growth condition, it can be a little higher. Means optimum can be 14 to 15 degree up to 17 degrees centigrade, and more than 20-25 degrees centigrade it's lethal to the, our growing species. Some of the suitable water quality that actually basically we consider during our trout farming. Most important is our dissolved oxygen is around more than 80% saturation, about 9 ppm, and also ammonia and total ammonia nitrogen. Ammonia is we know it is highly toxic species, so it is around 0.01 ppm, and up to 1 ppm we can the trout can tolerate total ammonia, that is basically constitute both unionized ammonia and ionized ammonia. And another one is total suspended solid. Because I have already discussed, it is side feeder, means it basically when you broadcast the feed to the tank or pond, directly consume the feed, because if the site will be not proper, means total suspended solid will be more than 30 or 50, 30 or 40, then it is very difficult the trouts to visible the feed, so the wastage of feed and also growth also will be affected. And also of course the temperature that I already told, around 12 to 18 degrees centigrade, it is very suitable for the growth of trout. Some of the hatchery and farming protocols, and that I was telling, two type of farming phases we can cover. One is full farming system that cover all life stages. Basically that aims from fingerling to market. From whole farming system, we do have some broodstock raising and maintenance, then selection of the male and female 1 to 2 months before, and sex-wise segregation, and rearing in different tanks, with giving good quality broodstock diet with ad libitum, then artificially dry steeping and fertilization, then incubation of eggs, then rearing of young one in nursery, finally raising fingerlings or rearlings, then raising the table size fish or broodstock. Some of the transportation, if we target the full system, then we do have some transportation and packing facility, because some of the eggs we can directly sell to the farmers, because nowadays farmers are very much means demand of the idova of the rainbow trout. So basically in from Champavat we used to transport the with this kind of systems. It is basically a box system. Lower layer will be this one ice pack will be there, ice layer will be there, then we can have the some of the this kind of some of these trays. In these trays the lower side will be small holes will be there, that the eggs will not means fall, but the some of the excess water will drop out, and over this we can cover, and we can have the some of the ice packs, and we can closely seal the eye. It can transport about 8 to 10 days also, it no problem. So from because in this system also foreign that Denmark stock I was talking about, we Denmark stock we transported from Denmark to our Bhimtar and also to Jammu Kashmir. So no mortality, or about 95, more than 95% survival is there. So small apart from this, and that we are actually generally transporting the iodova, but some of the farmers they need the ready to stock fishes, because if we transport some of the iodova, but the farmers do not have this type of hatchery facility to incubate the Idova, because Idova again it will take around 2 weeks or 10 to 12 days to become hatchlings. Because the rainbow trout, our incubation generally based on the degree days, and in FAO it is 360 to 370 degree days. Now, whatever the if suppose we are having 10-degree water temperature, then it will take around 36 to 37 days to be hatched. So for this purpose, some of the farmers they actually aim for stocking and aim for transporting the ready to stock stockable size. So for that we have conducted one small experiment. We have transported some one Patilikul farm. Patilikul farm is in Himachal Pradesh from Bhimtal, and so many permutation combination like some of the sedatives like our clove oil, then some sodium chloride, and packing density also alterations. Too many permutation and combination we have actually tried, and during the transportation we have double layer polythene bags with full of 1/3 full of water and 2/3 part is full of oxygen. In this condition we have transported, and finally we have the conclusion that while transporting these fingerlings of about 25 to 26 grams, if before than that 70 hours starvation with stocking density of 25 to 26 gram per liter biomass with sedation of clove oil with 40 microliter per liter, giving the 100% survival. Because we have transported from Patrikwal to Bhimtal, there was no mortality during the transportation process. And generally, the unpacking steps, it is I think it is most valuable step to be followed while while transporting from other farms to our farm for this stocking of the fingerling size, because generally we do hurry during our stocking of our seeds, but we do have some of the mistakes that generally we encounter when we some of the procedures we need to follow when we transport to the seeds to our farm for stocking purposes. When the seed packets are being transported to our farm, don't open these seed packets so very hurriedly, and do not decant all the material seeds to our tanks. It is better first half an hour or 45 minutes we should have kept the packets inside our grow out tanks, and after half an hour little bit temperature and some of the water parameters will be normalized. Then we should pour. You can see we should pour some of the water little by little with the help of mops, and fill the polythene bags with water. Then we can dip the polythene bags inside the grow out tanks. So automatically the seeds that are being transported, they are escape out from the packets. Do not decant the seeds directly to the grow out ponds, otherwise the mortality will be there. Then now we'll consider some of the trout farm designs. In trout farm, there are basically three types of design. One is series connection, another one is parallel connection, another one is combination. In series connection, we can see that some from natural stream we are extracting water from the natural stream, and it will flow from one tank to another tank, and it will so go on. But in parallel system, the each tank get a separate inlet system, so it will directly take the stream water. So basically in this system, the parallel system having the good survival, because if the first tank getting the water quality is very good, but in the last tank the water quality is getting worse, so it will affect the mortality and growth also pieces. But in parallel system, we do have different set of facility, so we can directly take the stream water to our pond, to every pond separately. So the water quality deterioration and what are the growth and survival, it will be not affected so much. These are the some of the trout farm. This is the trout farm of Himachal Pradesh. Here one Balwar Singh Arkiwan, our progressive trout grower, is and there. He has around 21 to 22 trout farms. This is, you see the parallel connection, and this is some of the farms also, government farms in Himachal Pradesh, and this is the some requested flow, means what are the exact amount of flow, or what is the amount of flow that we can have the better quality of fishes. Because you see this type of flow, nothing, the debris or some of the leftover feed or some of the fecal matter will wash out with this kind of flow. And during our this is our Champawat farm, and this is having some of the, for protection purpose, some of the bar fencing and snake or rodents used to enter the farm. This is to have some of the health issues to fishes, also they directly consume the fishes. So what are the consideration during the ratio flow through system? Generally, we do consider actually the ratio size of 30 to 45 meter square. It is having optimal, having 15 meter length and 2 to 3 meter this breadth will be there, and 1 to 1.5 meter height will be there. If 1.5 meter height will be there, then some of the our head space, some of the head space will be there, buffer space for the 0.3 to 0.4 meter buffer space will be there, so that the jumping or escaping of the fishes can be prohibited. And during the construction of our farm, our tanks, about 5 to 10% of the bottom slope is needed, so that the uneaten feed, metabolic waste, and other debris can be washed out properly. And in India, presently, generally we are following about the traditional system, about 20 to 30 kg per meter cube productivity, and total production is around 1500 ton. For this stocking, for this production of the rainbow trout, so generally we do prefer, the farmers do prefer the stocking of the fingerling size, because in fingerling size there will not be any such mortality, about 95% survival up to the table size species. So fingerling size we generally stock up to 100 numbers per meter cube with 10 to 20 gram size, and flow rate is generally maintained 3 to 5 liter per second, which is around 150 to 200 liter per minute. And within the thermal regime, it is 13 to 18 degrees centigrade. And in rainbow trout, unlike carps, rainbow trout is cannibalism, and it is a carnivorous fish, and it is a cannibalism in nature. So when we are growing the fishes, some of the fishes act as a shooter, so they grow fast. So in between every 15 days to 1 month interval, size grading should be there. And in our BCFR now, it is working and also succeeded about 20 to 25% better growth if we culture with triplets. And the harvesting also can be done within 10 to 12 months, having the 300 to 400 gram marketable size. Some of the you can see some of the field observation of the inlet and outlet management. Here you can see two types of channels is there. It is one of the farm in Himachal Pradesh, because it is he is taking the stream. It is not spring fed. So when the rainy season comes, this generally other than rainy season, directly the water entered into his farm. But during the rainy season, due to the siltation load, he made some of the our inlet system some zigzag manner, so that some of the sediments it can settle down in the in this inlet system itself, and the clear water can be entered into the farm. And This is some of the outlet arrangement. If because this kind of sluice gate design you have to make for the rainbow trout farm, because some of the uneaten feed and everything needs to be some of these kind of fluids in between they is to lifted, and some of the that fecal matter and all the debris are to wash out. For as we basically know, rainbow trout basically based on two things. One is water quality, another is good quality feed. So we need to have good water quality and having good basically good diesel oxygen and temperature thermal regime. So in our Champabad farm, in the storage tank itself, we have some of these splashers so that aeration can be done, and the quality of the water can be improved. And also in grow out tanks also these splashers are fixed to have the better water quality. And some of the indigenous things, because farmers do not have some of the specialized filters, specialized things, so indigenous things, we can have some of the input system having some cloth or bags that can have the trapping of the some of the our silt load. Then simply we can have some bottles, 2-3 bottles, and then a hole so that it will be sprinkled throughout the tank, and the DO and water quality will be improved. What is the importance of the water quality? The water quality, if already told, because it is basically a side feeder and carnivorous fish. So if good quality water has the good vision, so it can take the artificial feed very properly, and good quality water also have the high DO content. So the growing species, the respiration will not be affected. But in contrast, if there is having some impairment in water quality, these obnoxious gases like ammonia and carbon dioxide will be more in that system. It will automatically hamper the health of the species. And if you can in the farm, you can encounter this kind of problem if some of the fishes are not evenly swimming or evenly distributed throughout the tanks, they are congregated near the inlet system, then there should be some kind of water problem or sign of water shortage. These are the health and hygiene also need to be followed in our these are the pictures of our Champawat farm, periodically drying, then also cleaning of the tanks, then 5 to 10 ppm for cleaning of the tanks floor for the with camanophore, and also our nets and also accessories, farm accessories, while we are stocking or feeding or remove out the fishes. This kind of hand nets and also drag nets we are using. So these are need to be cleaned periodically. What are the do's and don'ts? What are the needs to be followed? That I was told earlier that cleaning and washing of the reservoirs and adequate water should be come into the reservoir for the better survival and growth of the fishes. Stocking of the healthy uniform size fishes. If size differentiation will be there, then acceptance feed will be also differentiate from the healthy ones and also weak ones, and weak ones generally gradually becomes weaker and weaker, and it will die, and or otherwise some disease problem is there, it will affect all the stocks. So appropriate stocking density need to be followed, and periodic grading need to be followed, and also in between one each crop some of the tanks should be cleaned and dried properly, and during our culture duration we should have protein rich diet, because rainbow trout is a some sea based or what we can salmon based fish. So it will require generally more than 40 to 45% diet. Unlike carbs, we can have some 25% food protein also. It is sufficient for the carbs. And along with this, some of the pelleted feed also, some of the extruded pelleted feed with the good quality nutrition can be supplied to the farm. farm fishes, and also in between some of the stocks which are having a little bit problem, or the sign of some abnormality seen, then in field way we can have some 10% commercial solution also we can be treated with them. Then of course some profitability measures need to be here, and after the culture of this species, when we go for some cold chain or transport to far distance from our farm, it need to be degoted and ice packing after harvest. These are some of the culture system for nursery rearing, nursery tank, grow out tanks, and also broodstock tank. And FAO standard is there. Means 1 liter per minute per kg if we are culturing without aeration, and if having aeration, then 0.3 liter per minute per kg of water flow should be there for our rainbow trout farming. And in the nursery tank, generally we do prefer 100 to 200 numbers per meter square, and broodstock tank generally we do not consider as number, we generally consider as kg biomass, 1.5 to 2 kg per meter square. For 2-3 months prior to breeding, we can stock separately during the broodstock unit. Some of the these are the flow through systems. Earlier I was discussing about flow through systems, but how can we improve our productivity within the same area? So our directorate is working on the RS system, recirculatory aquaculture system. This is some experimental aquaculture unit, RS unit we have developed in our directorate, and besides this, one commercial scale system also we have developed. And also now we are producing the rainbow trout, and also transporting to of the high-end hotels, and also local peoples also, they are also taking fish from our directorate. So for RS unit, RS basically a closed system unit, means from one point water enters, and it flows all overthrow our culture system, and again through a series of filtration and disinfection system, again it entered to the system. So for RS unit, these are some basic requirement. We do have some of the sediment, or in indigenously. For commercial system, we do have some high quality units, this kind of units. But for our indigenous system, we do have some of the indigenous design also. Some plastic barrels also we can use for our sedimentation system. And for drum and screen filter we do have, because it will filter the particles of about 30 to 60 micron, and also it will have automatic backwash mechanism. Some of the protein fractionation or protein skimmer or compensation need to be implemented, because it will remove less than 30 micron particles, so that clear water, our culture system get the clear water. And some of the biological filters, because in rainbow trout farming we give high protein feed, so basically nitrogen and ammonia excretion will be more. And for converting this ammonia, nitrogen to nitrate, and then some of the our denitrate, which the Pseudomonas species or some other species are there, so they can nitrate, they can convert to elemental nitrogen, and it can be escape out. And we do have also some heater and chiller. Heater for our extreme cold climate, and also chiller during this summer season. And in flow through system, generally we consider as aerator, it is sufficient for the flow through system. But for the RS unit, because we do have high stocking density, so for RS unit, oxygen cone or oxygen concentrator, oxygen concentrator should be implemented, unlike the conventional aeration, which is having 5 to 10% efficiency. Because you do have any query, means why aeration having 5 to 10% efficiency? Because we have calculator also, because generally in aeration we bubble it in the atmospheric air, and atmospheric air around 79% nitrogen, and about only 20% oxygen is there. So in 20% oxygen, we are entering into the system, but we are thinking it is 100%. But actually, we are entering 20% oxygen of the into the system, and in this 20% oxygen, all the oxygen are not dissolved in the water, because when we bubble the system with aeration, most of the oxygen, most of the airs are escaped out. So it is of generally very low use. It will increase the efficiency of DO efficiency of about 5 to 10%, but not more than that. So for RA system, this oxygen concentration concentrator, along with nano oxygen bubble system that we are also implemented in this our RA system, we have around 90% efficiency, because the DO that we generally in nano bubble oxygen system, directly we injecting directly oxygen, not the air. So, and in the nano bubble system, it in the water column, it generally resides up to 12 hours to 30 days. So it can increase the water up to 500 or 600% efficiency, or up to 60 ppm. And also we do have also ozonation and UV light for killing some of the our harmful bacterial agents. And for our in our RA system, normal elevation generally support about 40 kg per meter cube. But for oxygen cone with nano bubble system, we have already experimented. It can support about 120 up to 120 kg per meter cube. It is about 3 times more than the conventional system. And in conventional system, generally we do stock about 100 to 150 number. Sorry, we do stock about 15 to 100 number fingerling per meter cube. But in RA system, we can stock about more than 200 fingerlings per meter cube. And during our flow through system, we can harvest 300 to 400 grams size in 10 to 12 months period. But in our RA system, in 6 months of culture, we can harvest about 400 to 5 gram size. So 2 crop can be harvested in a year. Some of the rainbow trout cultures, these are in the Himachal Pradesh. These are some in Nepal. They are also culturing. Because in you might be surprised why this type of system they have constructed. Because we are living in plains, so we do not have the scenario of the hill region. In plains, we do have the ponds of some acres or hectares, but in hills, it hills around 10 meters square. Some farmers have around 10 meters square or 20 meters square of land, and also in uneven size and shape. So wherever water source is there, they have constructed this type of system so that they can culture the rainbow trout. These are the system. It is called. We have seen in Nepal, and this type of system also in some of the traditional farmers in Himachal Pradesh. And some of the farmers of Jammu Kashmir, they have constructed a good quality of raceways, and the farmers that I was talking about, along with the fish growing, they are having the angling and sports fishing facility, and also home fishing. So it is giving a wholesome means approach for the rainbow trout farming, and also revenue generation for their livelihood. Now we'll talk about the rainbow trout feeds. Before now, actually, in our directorate, we have developed some of the starter feed and grow out feeds. But before that, generally, farmers used to give some of the powder feed, which major percentage will be the fish meal, but the powder feed has the uneven size, so water quality pollution will be there, and also in case of egg yolk, also some farmers were giving during the early stage of their hatching, and some of the our commercially fingerling feed and grow out feed also they are giving as a trout feeding. But now our directorate has developed some rainbow trout feed with good quality and nutrition for their feeding. Because rainbow trout generally they prefer or they need rather they need about 40 to 50% protein based on their life stages. Yes, during their early stages they require more protein than lipid, but during the later stages, during the advanced size or table size species, they need less protein than the early stages. But during the broodstock, if we will raise for broodstock, the lipid quality will be more, lipid quantity and quality will be more. And some of this starter feed and grower feed we have developed 0.8 mm, 1 mm, 2 mm, 4 mm, now 6 mm also it is available. And these are the some of the ingredients we are generally means incorporating during the preparation of the feed. Now you can see the fish meal will be the more percentage in the rainbow trout feed. Now our director is working on the finisher feed. Means finisher feed after the your harvesting size is achieved, then we can have the some consumer preference also with the incorporating some of the finisher feed, so that the fresh quality could be improved, the nutritionally and also in the visually also. You can see these are the conventional feed that what is the means muscle appearance, and this is the pinkish appearance. If we are giving some of the pigments and not more than 1 month, if 1 month before our harvest we can have the some finisher feed, then it can improve the flesh quality. And of course broodstock feed 45, and also broodstock feed also we are working generally starter feed and grow out feed we have developed, and it is also commercialized, and it is in tie up with Growell. Now Growell is supplying in the broodstock grow out feed and starter feed to the required farmers. Now we are actually concentrating on the finisher feed and broodstock feed. And this is some of the field observation that I was in Champavat. That time some of the during our because this is our Champavat. This is actually one hatchery is there having 1.5 to 1 to 1.5 lakh idua capacity. So during some of the cases, like during this season particularly in rainy season, some of the broodstock have some problem. So we in field level we incorporated some of the vitamin and some of the anthrophlora and vitamin C, and this is the perfect ratio that we work out that giving the better result for the survival of the broodstock. So along with the addition of the some of the vitamin mineral mix. During rainbow trout feeding, the feed need to be, of course, stored in a proper place because it is having high quality, high lipid and high protein. So it is onset of rancidity and some of the wild animal, some rodents and they also take the feed. They also have some fungus. Some of the fungus also might take during the rainy season. So for this we can store the feeds in proper place, and during our starter during the feeding of our fry or spawn or fry, we can have some soils hand feeding, and also some of the our for grow out feed we can have some of the bigger size of the broadcasting some of the units. And also demand feeder, automatic demand feeder also we can implement, because demand feeder some of the trout farmers in Jammu Kashmir and Himachal Pradesh, they are having very large area, so manually feeding it is very cumbersome and very tired process. So demand feeder can be implemented, and but in demand feeder it is seen because it is constitute some localized area. If in demand feeder, we have seen most of the healthy fishes, they become healthier and grow more, but some weaker fishes, they could not get the feed, and also they have some mortality or some of the health issues. And during some obvious sign of feeding problem, if some feeding problem is there, then the cannibalistic, because there are some cannibalistic nature, so they are preying of fins, some they can cannibal one after another, one another. So this kind of system need to be avoided. And during the feeding of the fishes, as and the size of the fishes increases, the frequency decreases, and also protein content maybe decreases. And some of the record keeping, simple record keeping things we can have in our farm: simple store register, how much feed is taken into the store and how much it is adjusted, like that. Some of the farm register, how much quantity we have stock, and how much fishes are there, and a simple feeding register, because how many times in a day we fed, how much percentage we fed, and simple stock register and mortality register, how many fishes are sold and how many fishes are nearly recruited, and how many fishes are mortal, are dead during this culture period. And monthly balance, at what time, and how many percentage of fishes are graded, and what is the growth. The overall we can have some of the register during for our farm maintenance. And we can see the nutritional significance of rainbow trout. Rainbow trout is the fish having more percentage of protein. You can see 40% of the protein in the flesh. You can have 40% of protein about, and vitamin A, vitamin D. Generally, we consider vitamin D. Generally, it is synthesized in our body with the help of sunlight. But in you see, the rainbow trout is having more than 158 percentage vitamin D and some of the minerals, and also more efficiently, this is some highly unsaturated and polyunsaturated fatty acid that also we can have, say, in that rainbow trout place. And so some of the health benefits, we can have, because it have omega-3 fatty acids, so improve the brain and also heart diseases, and some of the arthritis, some of the joint pain. These also can be improved with the help of rainbow trout. Then this is the rainbow trout is the basic fish we have cultured in our cold water region, in high hill region. Another one is brown trout. Brown trout not yet it is cultured. It is in because brown trout having some of the parameters need high quality, but the growth is generally less than the rainbow trout. So people, farmers generally do not prefer the brown trout, but for the angling and sport feeding purpose, they can have some of the natural streams, and we can they have some catchment area there also. They have conducted some angling and sport fishing activities. And then another group is snow trouts. These are actually indigenous species I was talking about earlier. But in our region, these mainly 5 species are available: Progastos, Plagiostomus, Richardsoni, Niger, and Liviatus. Contributed mainly capture fisheries, and sold in local market as Pahari Machli, or you can see the small regional fish variety with higher price. Growth is not encouraging, but can be cultured with herbivorous species like carp in cement tanks, because in unlike our plain region like Odisha, Andhra Pradesh, we do have bigger systems for culture of the carps, and also we do have earthen ponds. But in hills, it is a problem having seepage, because in silt and sand quantity will be more in that in hill region. So generally, car ponds also which constructed with some polyline tanks, or also in a cemented systems. So in the cemented system, some of the 20 to 30% of these species can be stocked without no additional feed. So it can grow, and it can have some additional income to the farmers, and also for it has the quality, because it has the browsing habit. So it has the quality of cleaning the attached algae from the cemented tanks and systems. And of course, this is the golden mohasil. This is the most valuable sport fish in India, and in our Champavat area, and also in Bhintal area, some of the streams like Kali, Sariu, Ladhia, some of these streams, these are quite available, and people generally prefer with sport fishing and angling. and in DCFR effort, because earlier we do have a one Mahasiri hatchery at Bhimtal, Golden Mahasiri hatchery, but earlier we used to do, and people used to do the Mahasiri breeding. Mahasiri breeding is a tedious job. People used to catch the mature brooders, male and female, from the lakes, and they used to strip. But after a research of 8 to 9 years, it is the north cell of research I am reflecting here. After the research of 8 to 9 months, we successfully breed the golden mahasi in our captive condition. In that, the male female that we are growing in our tanks, it can be segregated 3 to 4 months for breeding. Then one major photothermal, it is carried out around 4 to 5 years of research, and we concluded 40 hours of light and 40 hours of darkness with temperature regime of 23 to 25 degrees centigrade. It can be matured. But one thing is that when during our maturation, we give generally in tanks, in cement, in FRP tanks, we do give the substrates, and this substrate, while the spontaneous spawning, the eggs are high in the crevices, which is very difficult to recovery. But after that, we have seen that we then put some muslin cloth. Then also the mahasi do not beat due to some reason. Then we have seen with substrate and GNRI injection, then stripping. This is a well-proved technology. We can have the 100% survival of the brooders, as well as 100% recovery of the eggs. Another one is neosucculus hecagnolysis. This is called as chocolate mahasi. This is also we have achieved with substrate input the breeding of these species. These are the species generally found in the some high regions of northeastern region like Meghalaya, and they constructed some of the mahasi sanctuary where they restrict the people to catch these type of fishes. And but this type of fishes generally not came to the culture practices. Of course, it is substantially incorporated in the culture system, but not as a monoculture system. And some of the exotic cars, we know that is due to this thermal regime of our hill region, the IMCs could not survive during the cold times, because in cold time it becomes 3 to 4 degrees centigrade also. So in that some of the valley regions, some of the exotic carps like grass carp, silver carp, and common carp, they are being cultured, and they are cultured in some of the polyline tanks, some of the cement tanks, and they are also cultured in some integration with the duckery, along with some of the chicken, along with some rice farming, rice field farming also going on in some of the Arunachal Pradesh. And this is our DCFR report. Actually, the existing strain that we know, the Bangkok strain of common carp, it is having problem of mature within 6 to 7 months, and so that the due to this effect, we do not have the growth rate of this type of species. So we imported 2 varieties of common carp from Hungary, improved variety of common crop, and we distributed across the country, some of the hill farmers of the country. And this is Rabcifs scaly, and another one is leather carp. This is mirror carp. This is Felsosonogi mirror carp. It is encountered about more than 30% growth rate than the existing strain. And then coming to some of the minor carps like Bangana Dero. Bangana Dero is the breeding and seed production technology. It is being standardized in our directorate at Bhimtal, and it gives around 1.5 lakhs per kg body weight. And like carps, it is having thermal regime of 20 to 25 degrees centigrade. It can breed very easily. I'm not going into the details, but these are some of the carps we have bred in our system, in our director have achieved for the seed production and breed technology, and farmers also incorporate that this type of carps like Bangana Dero, Bangana Debdebi, and Lebio, Dehachilus, these are incorporated as in combination with the some of the major carps. Like I was telling, some of the labiodictylos, these are also bred in our safer condition, and also because these kind of fishes, one benefit is that these are some herbivorous fishes and also algae eaters. So if we are stocking in our some cemented systems or our some polyline tanks, it will not only additional feed is not required, but if we are stocking with some of the exotic major carps, additional feed generally not required, and also it will clean our ecosystem. Like that, some of our Pangana Devdevi, this is also one kind of minor carp, and one field experiment has been done in composite culture of Mangana Devdevi along with Ostomobelangri Pengua at a ratio of 50 to 50 50% 1:1 ratio in farmers pond at Manipur gives the better result. And some of the indigenous small varieties like Beryllius vandalensis, this is actually basically an ornamental variety. You can see the male and female, male having some pectoral plate, having some pinkish color, and also very vibrant color. And these kind of fishes as can be included as our small indigenous fish variety, as our ornamental variety category. Then another one is Chogunius chogunio. Chogunius chogunio, this is also basically one kind of carp, minor carp. This is one base spawner. This seed production and breeding also is standardized in our diet trade, and it is also can be cultured along with the some of the major carps. And another one is Gara Gotella. This is basically an ornamental variety also, and also some food fish. And this kind of fish generally used in the aquariums for the cleaning purposes, because it is suckerhead like our some of the exotic varieties, like some Plecostomus ultivicis that we call the crocodile fish. This is some exotic variety. In place of the exotic variety, we can use this kind of variety, Gara Gotella, Gara Lambda, Gara Rudra. This kind of varieties we can be incorporated in our ornamental category. And another one is Lebue pengusia. Lebue pengusia is a very high-end fish. This is generally found in our Assam and Meghalaya region, basically restricted to Assam. It is having high frequent fish, about 7.5 to 8 kg per getting body weight. It can give the egg of 7.5 to 8 lakh eggs per kg body weight. And it is now cultured in Assam, monoculture also, and also along with the some IMCs also. And another one is Sempelta. It is called a Japanese kingfish. This is one variety of minor carp. It is having some moderate size, not like the some major carp. So it can grow up to 250 or 300 gram. It is marketable size, 200 to 250 gram, and it is having price range of 700 to 800 rupees per kg. It can be sold. And another one is Ostomoma belangiri. It is a local fish of Manipur, and it's a state fish of Manipur also. It is generally known as Pengba, and one small experiment has been conducted with 10 farmers pond at Manipur Thobal in collaboration with our directorate, and with combination of some of the exotic carps like Rabcifs, silvercup, and Benga. So in final output is Pengba with grass carp and silver carp with 40:30:30 ratio gives the better result. And along with this, some of the ornamental varieties. Now we are having some indigenous ornamental varieties in our local stream that we are standardizing the breeding and seed production technology, and also distributing to the farmers. But some of the wild variety ornamental like koi carp, goldfish, and oscar, these are also we are concentrating on this. This is the indigenous ornamental fish, Crossotelius latios. This is some Glyptothorax species. This is some loches, that is Sistura species, and This is dark massive. This is Nemaquila species, and some of the loaches like Botia almerae, Botia almerae, sorry, Gara anandalai, and some of the scissorid catfishes like Gyptotharas and some of the other catfishes also are now we are concentrating in our indigenous seed production and seed production technology in our diet. So, lastly, coming to what is the nutritional significance, as we know, fish, excellent source of protein with high biological value means and the other source of protein like our chicken or some pigs or beef, these are having high red meat content. These are high red meat content, also more fibrous protein not digestible by the humans properly. But fish has the good amount of digestible protein, and also some of the high unsaturated PUFA, that is high unsaturated fatty acid, polyunsaturated fatty acid, omega-3 and EPA, eicosapentaenoic acid, and docosahexaenoic acid. These are the good quantity of fatty acids also available. Omega-3 fatty acids also available in fish. Along with this, this is also rich in vitamin A, D, some of the vitamin B1, B3, B6, and trace minerals like calcium, phosphorus, iron. These are also plenty available. So one saying is there, fish rich food for the poor, because poor people also afford fish in our Odisha. You can see generally village peoples, poor peoples do not prefer this kind of vegetables, more in vegetables, because actually it is expensive also. But in fish, they can get very cheaper price also, and also their regular habit is nearby canal is there, or nearby small backyard pond is there. They used to catch the small fishes, and they used to have it, and also helpful for the affluent. Affluent means it is also nowadays people are concentrating on the health. So the rich peoples, they are also concentrating on the consumption of the fishes. And some of the Udia paper cutting that I am posting here for your information. So everywhere it is for it is Smaran Shakti Badhita, Dushti Shakti Badhita, everything. Most of the things are covered in our health. It is mainly due to the fish consumption. So basically, we insist generally people should take fish, especially the children also. They should prefer fish for their consumption. Thank you so much.
Good afternoon to all. It is a matter of great privilege to propose a vote of thanks on this webinar. First of all, I would thank Almighty God for making today's webinar a resounding success. I am extremely thankful to our resource person, Dr. Sulip Kumar Maji, for sparing his valuable time to enlighten us with his commendable talk. I also thank our speaker, Dr. Abhay Kumar Giri sir, for enhancing our understanding pertaining to the cold water species. My heartfelt thanks to our principal, Ma'am Sangeeta Sahu, for her support to conduct this webinar. I would also thank our organizing committee members, Dr. Dilip Kumar Bed sir, Taslima Begum, Mr. Biswarit Panda, who have worked hard to ensure this webinar a memorable success. I also owe my special gratitude to our computer section department staffs, especially Sanjoita ma'am, Manas sir, Jyoti Mahanta sir, who have taken care of audiovisual systems very nicely. Lastly, but not the least, I would like to thank all the academicians, researchers, faculties, participants, and my dear students for your patient hearing and making this webinar successful. Once again, I thank you all from the core of my heart for your participation.
Parents,